Hey guys, I'm back. This might be really awkward. I'm in Arcadia Cafe, which is a beautiful little place in Ames, right in Campus Town. But today they're actually roasting the beans, and I was hoping they would be done soon, but it doesn't look like they're going to get done anytime soon. But anyway, that's where they roast. Really, really cool little shop. If you ever need a place to study, this is a great place to do it. So just testing volume levels. Let's see where we're at. Hey everybody, I'm back. We are here again looking at memory, cognition unit 7A still. This is a really detailed unit. In case you're wondering, I'm holding the mic up because it's really loud in here. I'm in Arcadia Cafe and they are roasting the beans for their coffee that they make in-house. It's pretty sweet. They also bake a lot of great things too. So if you need a place to go, just head down Lincoln Way and Ames to Arcadia, right next to Campus Town. Great little place to do some work. So let's get started. Today we're going to talk about biological bases of your memory. So common misconception that all of your memory is stored in one place. People typically think that it's in the parietal lobe. Not true. Lashley in 1950 tests rats that he gave lesions to on their brains after they had learned how to run a maze. And he put these lesions in different places on each rat. So each rat had one, maybe two lesions, and they put the lesions on different parts of the brain for each rat to see if there was a certain part of the brain that housed memory. And they found that all of the rats, after the lesions were placed, were able to still traverse the maze. So despite having damaged areas, memory was only weakened, not obliterated. So memories are not located in only one part of your brain. You store memories in all parts of your brain. Now, different memories are stored in different places. For example, in your frontal lobe, that's more of your practical knowledge area, you're going to have a lot of process storage there. Your parietal lobe, you're going to have a lot of experiential storage. So the episodic memory that I was telling you about is going to be more in that parietal lobe. Let's talk about the biology side of this stuff. Now, this stuff is going to get pretty serious. A lot of terms here, but you should have already seen them all from your vocab sheet. So we already know that experience physically changes your brain. So when learning takes place, more serotonin is released at certain synapses of the brain, wherever the learning's occurring, basically. And this makes your neurons in the network more likely to fire. So sending neurons are more likely to fire, and then they release those neurotransmitters, and receiving neurons seem to increase the receptor sites at this time period while you are learning. It's kind of interesting. And if you do this over a long period of time, then you have what is called long-term potentiation. That means that your ability to fire that action potential process of the neuron is quicker, and it can happen more often, and this stays in the long term. So I've been in school for a decade, every single day, constantly learning, so I probably have quite a few more neurotransmitters to release in certain parts of my brain, wherever the learning has been occurring most often, and I probably have more receptor sites on the receiving neurons. Another thing to note here, which is really cool, we've talked a lot about Alzheimer's disease in our class, and we're going to continue to talk about it, but this unit really hits on Alzheimer's quite a bit. ACH, which is also known as acetylcholine, we've talked about before. Go back to Unit 3, Biological Bases of Behavior, you'll see it. It plays a huge role in memory. So basically, ACH allows us to keep those memories. Now, as you age, as you get older, you are naturally not producing as much acetylcholine. And when that happens, you can tend to lose some of your memories, but certainly not all. Alzheimer's patients, they've noticed a direct link with acetylcholine levels that drop by as much as 90%. This results in a significant loss of cognitive function and behavioral function. So we're seeing a huge connection there, and some of the research right now is figuring out how to increase those acetylcholine levels to prevent Alzheimer's. Okay, the impact of long-term potentiation. So there are drugs that block long-term potentiation. Actually, pretty much all of the ones that you know about block your long-term potentiation. And when your long-term potentiation is blocked, this interferes with your learning. There are also drugs that can enhance and increase memory ability. Think the movie Limitless. While that's entirely fictional, it does happen. There are certain drugs that like kids are taking in college to stay up later so that they can study longer, and these actually expand your long-term potentiation, but they're really harmful for you, so don't give it a try. Long-term potentiation inhibiting drugs can actually erase some of your recent learning. So say you go out and have fun with your friends in college when you're 21, drinking some alcohol the night before you have a big test. You are likely to lose some of that learning for the test. So memory enhancement, well, Krebs protein boosters may help trigger long-term potentiation. Certain foods provide for Krebs. Blueberries are the only one that I know of. Give that a look. You might want to check that out and see what that can do for you. Neurotransmitter glutamate may also enhance LTP. Not monosodium glutamate. That's not going to help you at all. That's a combination of sodium and glutamate, and so it just changes the whole chemical makeup of the thing. But glutamate itself, the neurotransmitter, may enhance LTP. And then electroconvulsive therapy and head trauma may disrupt your memory and learning as LTP in process is not completed. So basically, the learning that you're doing currently, if you were to suffer head trauma in this moment, the process would not be complete. You wouldn't remember what you learned. All right, stress and memory. This one I know all too well in my PhD program that I often refer to. Incredibly stressful test. You study for 3 years for this test, and you get 2 shots at it. And this test, there's a lot on the line here. Your funding, whether or not you get an advisor for your PhD work, all sorts of stuff. So studying for this test, you start developing stress. And really, instead of it being whether or not you understand the material, it ends up being, can you stave off the stress and learn enough in order to take this exam? So I had a serious problem with this, especially, living alone, and I'm not an alone kind of person. It just ate away at me, and I swear I would study all day and I wouldn't remember a single thing. So when we get excited or stressed, we produce more hormones that make more glucose energy for the brain. And this leads to increased activity in your amygdala and the limbic system, which is also involved in the formation of memories. Sometimes this can be an incredibly positive thing, as explained here. Other times it can be pretty bad. So strong emotions equal stronger memories. Weaker emotions equal weaker memories. That's why you don't remember the day-to-day drudgery of your life. You remember those significant moments. And this also helps us to explain flashbulb memory. If you think about the term flashbulb, it goes back to the times when we used to have those cameras with the big flashbulb on the top of them. And really, just take your cell phone and take a picture without the flash on, and then take a picture with the flash on. Everything in the picture with the flash on is more vivid and bright and detailed and distinct. And that's what flashbulb memory is you have this memory of something that is so incredibly vivid it feels real. When we usually don't remember things, that specifically. Also, our hippocampus and explicit memories. Your hippocampus helps a lot with your explicit memory. So it's responsible for transferring short-term memory to your explicit long-term memory. And prolonged exposure to stress hormones can actually shrink the hippocampus and inhibit memory, which is most likely what was going on with me during the PhD program. So damage can disrupt movement of information to the cortex, your long-term memory. And your cerebellum processes implicit memories and classical conditioning unconscious processes. So where are your memories stored? Well, here are all the different places that they can be stored. First, in that frontal lobe, we've got our semantic and episodic memories here, but you also have some of your episodic in your parietal lobe. Then your motor cortex is involved in storing procedural memories, obviously, right? Learning how to kick a soccer ball, procedural memory, motor cortex, frontal lobe. Then we move down to our cerebellum. This plays an important role in the storage of procedural memories as well. Hippocampus is pivotal in the formation of new long-term and semantic and episodic memories. So So without that hippocampus, you might not be able to develop long-lasting relationships because you would have no memories of them. Your amygdala is vital to formation of new emotional memories. So your first kiss, a nightmare, a car accident. Why can I think of more negative than positive ones? All of these things will be your amygdala. So your temporal lobe is involved in the formation and storage of long-term semantic and episodic memories and contributes to the processing of new material in short-term memory. This is also, you're going to have a strong association between sound and memory here, obviously, because your temporal lobe is also responsible for your hearing. And the prefrontal cortex. This is involved in the storage of your short-term memories. So memory is stored a lot of places, actually more than what is mentioned here. But this is a pretty good visual, and this is mostly what they'll be expecting from you on the test. All right, that concludes it for today. I'm going to put this retrieval in with forgetting in our next podcast. So that's all for now, gang. See you next